This is the video for Day 8 of the 2022 Short Read Workshop. In this video, we'll be talking about how to use more complex experimental design setups in DESeq2. Last time, you learned how to run a pairwise comparison in DESeq2, such as the example shown here. In these experiments, we created a metadata file, which designates our samples into one of two conditions groups. In this case, we want to compare two disomy samples to two trisomy samples. DESeq2 also requires a counts file where each sample is assigned a read count number for each feature. You can see one example of this feature drawn as a box plot to the left. The columns of the counts file are in the same order as the rows of the metadata file. Once we have this metadata and counts file, we can run the DESeq2 algorithm. In the last video, you focused on learning about the size factor and dispersion estimation steps. Here, we'll be focused on the model fitting and hypothesis testing steps. When we run the results command on our DESeq2 object, we obtain a list of genes which were compared in the two groups. DESeq2 also prints out a base mean, which is an average of the normalized counts, a log2 fold change, which is a value estimated from using a general linear model, and then a p-value, which is a result of the statistical test that was run on these genes, either a Wald test or a likelihood ratio test. But this setup was limited to only the most basic pairwise comparison, where our samples can fall into one of two groups. Today, we'll learn how to incorporate more complex design elements into DESeq2. Here are some examples of the designs we'll be exploring today. Here is a cartoon indicating a multifactor comparison between a treatment and a control in two different genotypes, genotype A and genotype B. Here, we'd want to figure out whether the treatment has a different effect between the two genotypes, which requires us to quantify the differences in their response and compare them. Or, our experimental setup may contain a double treatment. Here, we'd want to figure out if the two treatments behave additively or if they have a synergistic effect when used together. We may also have multiple groups within the same factor, such as cells that were gathered from different individuals from the same family. Here, we'd want to run pairwise comparisons between these family members, but there isn't a baseline or control sample here. Rather than run a series of different DESeq2 experiments, we'll learn how to keep them in the same run and easily generate these comparisons using contrasts. Finally, even within pairwise comparisons, non-biological factors, such as if two different scientists produce the datasets, can produce unintended batch effects. We will learn how to account for these batch effects to improve the fidelity of our analysis. The key to each of these designs lies in how we construct our call data, or metadata, and our design matrix. The design matrix is a simple matrix of zeros and ones which tell DESeq2 how to group the samples for our desired comparisons. You can run the model.matrix command along with the metadata, and a tilde followed by the columns you want to view from that metadata as a design matrix. This part is happening internally in DESeq2, but it's a good idea to check it manually to get the hang of these complex designs. You'll notice that the design matrix rows line up with the rows of our metadata data frame. The design matrix is interpreting the values in our conditions column to be either 0 for disomy 21 or 1 for trisomy 21. You'll also notice that all samples have an intercept value. DESeq2, and GLMs in general, construct a model by first establishing a baseline, or intercept, and then later establishing a difference coefficient relative to this baseline. For samples with a 0, this difference coefficient is multiplied by 0. As such, the only value left for the fit is the intercept. Essentially, these samples will only be used to establish our baseline. By default, DESeq2 picks the first alphabetical entry in your column as the baseline samples. In this case, that's disomy 21. You can manually assign the baseline by reorganizing your samples using the relevel function in R. the samples that have a 1 in this column will have a difference coefficient. In other words, the difference coefficient is multiplied by 1, and so the difference is added to the intercept to get our final fit for this group of samples. Finding this difference coefficient is what yields our fold change estimation. Essentially, DESeq2 is using the design matrix as a way to group these samples together, so it can find the lines of best fit for these groups. The distance between these lines are our coefficients, or the fold changes between those samples. The actual fitting process is beyond the scope of this course, but this is essentially all that's going on inside of DESeq2. Now that we understand the basics of linear models, we can talk about batch effects. If we just group the samples by their condition, as you learned how to do yesterday, we notice that there's a large effect for this gene between disomy 21 and trisomy 21 samples. However, you'll also notice that there is a large amount of variance within both of these groups. As a result, the genes look very noisy, and it can be hard to tell whether or not this is actually a significant effect. But let's say we had some more information about these samples. For example, if we knew that both of the replicates were generated by two different scientists. The fold change that both treatments observed is thus similar, shown by the blue arrow, but the baseline is different between the control samples. This is a batch effect, and potentially explains the variance that we saw. We can add batch information as a new column in our metadata data frame. This will instead group the samples by the batch that they were generated from. Then, we can add this effect as an element of our design matrix. When we run the model.matrix command, you can see that the samples are now grouped both by batch and by whether or not they are trisomy or disomy 21. In other words, we have a new difference column that instead looks at the effect that the batch has. Using this setup, we can account for the variance between the baselines of the two batches. We can now run DESeq2 as before, but with our expanded design. This new design, tilde-batch plus condition, will automatically account for the batch effects, and our results file will print out the updated calculations. Now we can compare the results here to learn a little bit more about what we just did. For example, if we look at the gene in the red box, you'll notice that it is considered significant when we account for batch effects, but it is not considered significant if we do not account for batch effects. This isn't because the fold change estimation is much different with either method, but because the standard error of that fold change estimation is reduced with batch correction. Essentially, we were able to explain away some of this standard error by using the batch information, which aided in our ability to call this gene as significant. I'll provide more sources for learning about the details of this fitting process tomorrow during class, but for now, just know that adding batch information as a term in the linear model is a way to correct for batch effects in differential analysis. Some more notes here about how DESeq actually performs this analysis. By default, DESeq2 is going to print out the last element of your design formula in your results table, but it saves all of these results within the DESeq2 object itself, and they are fully accessible at any time. For example, you can use the coef command on the DESeq2 object to see fold change estimations and the intercept values for all of the groups. And if you're interested in hypothesis test results instead for the other group, you can use a contrast. A contrast can either add or subtract coefficients for in or across group comparisons. In this case, for example, we could use a simple contrast within the batch group to compare B versus A. As you can see, when we print out our results table now, the results instead look at the groups B and A in the batch column as opposed to the condition effect like we were looking at before. This also works if your designated group has more than two elements in it. So, for example, if we've instead generated datasets from this family of individuals, we only have to designate the person the sample was generated from in the metadata file, as shown here. The design matrix will automatically be built to reflect which samples belong to each person, and we can then use a contrast to compare the samples from two individuals, such as Ethan versus Eli, as shown here. Here, we can see that DESeq2 results command gives a nice summary of the requested contrast. This is convenient as DESeq2 is able to calculate these comparisons on the fly, even if both have a difference coefficient, which saves you from having to rerun the comparison over and over again using the sample of interest as a baseline. In other words, getting the other results is easier if we just use a contrast rather than running the entire experiment again. Now that we've talked about those simpler comparisons, let's move on to multifactor analysis. Here, we're interested in finding out whether there is a combinatorial effect from our group designations. Shown on the left is an example gene and its signal in 4 different groups: a control and treatment in genotype 1, shown on the left, and a control and treatment in genotype 2, shown on the right. Here, we want to find out if this gene responds differently to the treatment in the 2 genotypes. I'm going to represent the box plots from the last slide as a cartoon here to keep things simple. Here's the metadata for the experiment. You can see that we have a control and treatment designation in the treatment column, and genotype 1 and 2 in the genotype column. But we can't just run a simple pairwise comparison between treatment groups because we're also interested in this other column. What we really want to compare is the difference between treatment and control in genotype 1 and the difference between treatment and control in genotype 2. What we need is an interaction term. To do this, we'll construct our metadata as shown here, designating each sample's batch, genotype, and treatment, and then construct our model matrix with an interaction term. We add batch, genotype, and treatment as before, and then a new term, genotype, treatment. The colon denotes the interaction between genotype and treatment. What does this matrix look like? Well, we can see that our expected batch, genotype, and treatment information is there, but there's a new column, genotype 2, treatment. This new column has zeros for every sample that isn't both treated and genotype 2. So only the last samples will have this coefficient. The other samples will have coefficients for either genotype or for treatment differences, but not for both. Thus, DESeq2 can separate out the interaction effect of the genotype and treatment from their individual background effects. Here's what that will look like in practice. We run DESeq2 as before with the expanded design matrix. Our reported fold change and p-values are now for the interaction effect by default, as it's the last term in our design matrix, although the other comparisons are available using a contrast. Now, we can draw out a visual diagram of exactly the process that DESeq2 just automated for us. Essentially, we fit a model using each of the parameters in the design matrix. For simplicity's sake, I'm going to leave out batch effects from this model. Let's take a look back at our design matrix data alongside this diagram. We can see the samples in the red box only have 1s for the intercept, while they have 0s for the genotype 2, treatment, and genotype 2, treatment columns. These will thus establish our baseline, shown here in the bottom left. The samples in the blue box have 1s for the treatment effect, so they'll have the difference coefficient for this treatment effect, indicated by the light blue arrow. The brown box samples have 1s for the genotype, so they'll have the genotype effect, indicated by this brown arrow. But then something interesting happens. For the samples in the green box, we calculate the treatment effect in genotype 2, shown as the dark blue arrow here, but we don't stop there. The interaction term is calculated by taking the treatment effect in genotype 2 minus the treatment effect in genotype 1. In other words, we take the dark blue arrow minus the light blue arrow, which yields the green arrow. This green arrow can be interpreted as the genotype-specific response controlling for our baseline effect. I want to make one more note about this process before we move on. We set out to answer this question: is the treatment effect different across genotypes for each gene? That means what we're actually testing here is a null hypothesis that the interaction effect is 0. In other words, that the light blue arrow is equal to the dark blue arrow. Since the dark blue arrow is clearly much different than the light blue arrow for this gene, the resulting interaction term, the green arrow, is very large, and thus this is a significant gene call as indicated by the p-value. But for other cases, this might not be what we're interested in. Maybe, for example, we're interested in just the treatment effect for genotype 2. Put another way, maybe we want to just look at the value of the dark blue arrow. This is accessible using another contrast, this time using a list. Instead of comparing within a group, this list allows you to add or subtract group effects. So we can add the main treatment effect, treatment versus control, and the interaction effect, genotype2.treatment. Put another way, we add the green arrow and the light blue arrow to yield just the dark blue arrow. Appropriately, you can see the log2 fold change value is smaller than before, as this arrow is shorter than the interaction term. Now, some of you watching may have already thought of another solution for getting this blue arrow. We could instead reorganize our data, set genotype 2 as the baseline, and rerun the experiment. The main treatment effect would then be calculated as the blue arrow, and we wouldn't need to use a contrast. And this is true, you can reorganize your data frames to get these effects. Your choice of baseline will inform how you run these experiments, although it should be noted that this does require you to rerun DESeq and refit the whole model. Here's one more very common experimental setup. If you're feeling confident, pause the video and try to figure out how you would construct the design matrix to test the interaction effect of the two treatments. You may have noticed that we can essentially treat this experiment the exact same as the last one. That flexibility is one of the major benefits of linear models in general, not just here in DESeq2. We can just assign the columns for the batch, presence of treatment 1, and presence of treatment 2, as shown here on the right. You can then just use those terms in your design matrix, including an interaction term between treatment 1 and treatment 2. I want to talk about one more bonus design that you may encounter. Depending on how efficient we are in class, we may or may not be able to go over this together. Some experiments use serial information in their design. In this case, we may have many data points which are related by a common factor, such as time, concentration, age, or some other continuous variable. Imagine here, for example, that we treated control and mutant cells and then harvested RNA for sequencing at 6 hours, 12 hours, and 24 hours. We have an untreated control at 0 hours as well. We have two choices for how to test these data. First, we can run a series of pairwise comparisons using contrast, which you already know how to do. For example, we can control 6 hours and 0 hours in the control and mutant samples, 12 hours and 0 hours in the control and mutant samples, and 24 hours and 0 hours in the control and mutant samples. Of course, we could also change our baseline to be another time point to get the other comparisons, such as 12 hours versus 6 hours, or we could get this by using a contrast in the other experiments. But this doesn't utilize the true power of a time series analysis. What we'd really like to do is test the whole dataset at once by fitting a model to the whole thing. In this method, we'd fit two different models, one full model with all of our parameters, and one reduced model, which leaves out the element that we want to test for significance. For example, in the red and blue lines, we see model estimates, which are informed by both the time and by the genotype of the sample, whereas the purple lines indicate a model which is only informed by the time of the sample. Genes that are better explained by the full model would be our significant calls. In essence, all we're doing here is assessing how well each model does at explaining the data. A full model with more parameters will always do at least as good or better than a reduced model missing some of those parameters. So to test whether a parameter is truly adding a significant amount of the model's ability to predict the data, we compare a full model to a reduced model in what's known as a likelihood ratio test. Likelihood here refers to the probability of observing the data given the model parameters we constructed. To visualize a likelihood ratio test, you can imagine it like comparing the distance from the red and blue lines to their dots in the last slide to the distance of the purple line to the dots in this slide. Now, likelihoods are actually calculated from a probability distribution, which usually aren't just flat lines as I've depicted them here. So the calculation for the likelihood ratio test is more complex than this diagram would indicate. We won't get into these details during the workshop, so I'd recommend reading up on likelihood and model fitting if you're interested in a more in-depth explanation. One more note. I've depicted the model here as a series of horizontal lines fitting each category, but a model could also include more complex fits, such as a linear regression or a polynomial for continuous data. You can compare the likelihoods of the models in the exact same way. If your data calls for this type of comparison, though, you should consult with a statistician to help you set up the generalized linear model. For our example here, we can set up our metadata table exactly as we did before, with batch, genotype, and time information listed for each sample. From there, it's just a matter of picking our full and reduced models. We designate our full model in the design matrix as before, and then we change the test parameter to LRT when we run DESeq. We'll also need to include a reduced model. The elements that we leave out of the reduced model will be the ones that we are testing. In this case, I've left out the genotype and the interaction term. You'll notice that even though we're comparing across the entire time course, there's still a reported log2 fold change and log fold change standard error value. The fold change and the standard error value are just a default printout from the last coefficient that we put into our matrix. So you can go ahead and ignore these here, as they don't make sense in a likelihood ratio test. What we're mostly focused on are the stat and p-value numbers here. As a rule of thumb, whatever terms aren't in your reduced models are the ones that you're going to be testing. So, if you only include tilde time, you'll be testing the effect of genotype. And if you only include genotype, you'll be testing the basal effects of time. It helps to sketch out exactly what effects you'd like to test, and then ask yourself which elements belong in the full and the reduced models, using a little diagram or a cartoon like I've done here. And with that, I hope that you've understood the basics of general linear models in DESeq2. Tomorrow we'll be going over how to actually do all of this in R and DESeq2 using some real data.